Good morning, students. Today I am teaching you main characteristics of growth and regeneration. This topic, main characteristics of growth and regeneration, is included in the paper 3rd Developmental Biology of BSc Part 1st. Students, in this topic, we will study the growth and regeneration process during embryogenesis and during the lifespan of organism. Student, tissue growth involves both an overall increase in tissue size and also the differential growth of parts, allometry, which contributes to morphogenesis. Growth mostly occurs through cell proliferation but also through changes of cell size or the deposition of extracellular materials. So, students, growth, the increase in cell size and number that take place during the life history of an organism. Now we are going to study the main characteristics of growth. Growth is seldom random, rather, it occurs according to a plan that eventually determines the size and shape of the individual. Growth may be restricted to spatial region of the organism, such as the layers of cells that divide and increase in size near the tip of the plant shoot, or the cells engaged in growth may be widely distributed throughout the body of the organism, as in the human embryo. In the later case, the rates of cell division and of the increase in cell size differ in different parts, that the pattern of growth is predetermined and regular in plants and animals can be seen in the forms of adults. In some organisms, however, notably the slime molds, no regular pattern of growth occurs and a formless cytoplasmic mass is the result. So, students, the rate of growth of various components of an organism may have important consequences in its ability to adapt to the environment and hence may play a role in evolution. For instance, an increase in the rate of growth of fleshy parts of the fish fin would provide an opportunity for the fish to adapt more easily to terrestrial locomotory life than could a fish without this modified fin. Without disproportionate growth of the fin, ultimately resulting from random changes in the genetic material, the evolution of limbs through natural selection might have been impossible. So, the students, now we are going to study the growth in cells, the increase in size and changes in shape of a developing organism depend on the increase in the number and size of cells that make up the individual. Increase in cell number occurs by a precise cellular reproductive mechanism called mitosis. During mitosis, the chromosomes bearing the genetic material are reproduced in the nucleus, and then the doubled chromosomes are precisely distributed to the two daughter cells, one of each chromosomal type going to each daughter cell. Each end of the dividing cell receives a complete set of chromosomes before the ends separate. In animal cells, this is a pinching off of the cell membrane. In plant cells, a new cellulose wall forms between the new cells. During the period of cell preceding the actual distribution of chromosomes, the mother cell often grows to twice its original size. Hence, a cycle consisting of cell growth and cell division is established. Cell growth. An increase in cytoplasmic mass, chromosome number, and cell surface is followed by a cell division in which the cytoplasmic mass and chromosomes are distributed to the daughter cells. An increase in cytoplasmic mass does not always occur during cell division cycles. However, during the early development of an embryo, for example, the original egg cell, usually a very large cell, undergoes repeated series of cell divisions without any intervening growth periods. As a result, the original egg cell divides into thousands of small cells only after the embryo can obtain food from its environment. Does the usual pattern of growth and mitosis occur? Now, students, we are going to study the growth in animals. The growth of animals is more restricted in time than is that of plants, but cell division is more generally distributed throughout the body of the organism. Although the rate of cell division differs in different regions, the capacity for cell division is widely distributed in the developing embryo. Increase in size is rapid during the embryonic period, continues at a reduced rate in juveniles, and thereafter is absent. Cell division and size increase continue, however, even after increase in total body size no longer occurs because these events are balanced by cell death. Post juvenile increase in cell number is primarily a replacement phenomena. Height increase in mammals is limited by cessation of cell division and bone deposition in the long bones. The long juvenile period of growth in human is unusual. Most higher animals attaining mature size soon after the end of embryonic development. Some organ systems undergo little cell division and growth after birth. For instance, all the germ cells, precursor of egg cells of the female, are formed by the time of birth. Similarly, all of the nerve cells of the brain are formed by the end of the embryonic period. Further increase in the size of the nervous system occurs by outgrowth of nerve fibers and deposition of a fatty insulation material along them. Although the greatest increase in size of nerve cells occurs as in plant cells, after the cessation of cell division, the nerve fiber outgrowth in animals represents a true increase in the amount of cytoplasm and cell surface and not just an uptake of water. So, students, some organs retain the potential for growth and cell division throughout the lifespan of the animal. The liver, for example, continues to form new cells to replace senescent and dying ones. Although cell division and growth occur throughout the liver, other organs have a special population of cells called stem cells that retain the capacity for cell division. The cells that produce the circulating red cells of mammalian blood are found only in the marrow of the long bones. They form a permanent population of dividing cells, replacing the red cells that continuously die. and disappear from the circulation. The rates of both growth and cell division can vary widely in different body parts. This differential increase in size is a prime factor in defining the shape of an organism. So, students, now we are going to study the process of regeneration and how it occurs in the body of organism. In biology, regeneration is the process of renewal, restoration, and tissue growth that makes genomes, cells, organisms, and ecosystems resilient to nature fluctuations or events that cause disturbance or damage. Every species is capable of regeneration from bacteria to humans. Regeneration can either be complete, where the new tissue is the same as the lost tissue, or incomplete, where the necrotic tissue comes fibrosis. So, students, at its most elementary level, regeneration is mediated by the molecular processes of gene regulation and involves the cellular process of cell proliferation, morphogenesis, and cell differentiation. Regeneration in biology, however, mainly refers to the morphogenic processes that characterize the phenotypic plasticity of traits, allowing multicellular organisms to repair and maintain the integrity of their physiological and morphological states. Above the genetic level, regeneration is fundamentally regulated by asexual cellular processes. Regeneration is different from reproduction. For example, Hydra perform regeneration but reproduce by the method of budding. The Hydra and the planarian flatworm have long served as model organisms for their highly adaptive regenerative capabilities. Once wounded, their cells become activated and restore the organs back to their pre existing state. The chordata, urodales, salamanders, and newts, an order of tailed amphibians, is possibly the most adept vertebrate group at regeneration, given their capability of regenerating limbs, tails, jaws, eyes, and a variety of internal structures. The regeneration of organs is a common and widespread adaptive capability among metazoan creatures. In a related context, some animals are able to reproduce asexually through fragmentation, budding, or fission. A planarian parent, for example, will constrict, split in the middle, and each half generates a new end to form two clones of the original. So, students, in echinoderms such as the sea star, crayfish, many reptiles and amphibians exhibit remarkable examples of tissue regeneration. The case of autotomy, for example, serves as a defensive function as the animal detaches a limb or tail to avoid capture. After the limb or tail has been automized, cells move into action and the tissues will regenerate. In some cases, a shed limb can itself regenerate a new individual. Limited regeneration of limbs occurs in most fishes and salamanders, and tail regeneration takes place in larval frogs and toads. The whole limb of a salamander or a triton will grow again and again after amputation. In reptiles, chelonians, crocodilians, and snakes are unable to regenerate lost parts, but many kinds of lizards, geckos, and iguanas possesses regeneration capacity in a high degree. Usually, it involves dropping a section of their tail and regenerating it as part of a defense mechanism while escaping a predator. If the predator catches the tail, it will disconnect. So, students, thanks for listening and thanks for your patience.